Okay, so a pleasant good afternoon, everyone. And today we'll be looking at reproduction. And when you think about reproduction, the importance of reproduction lies in the fact that it is critical. It prevents us from going extinct as a species, which is why in terms of the body's expenditure of energy into creation of approximately 150 million sperm per ejaculate as it relates to the amount of semen expended during the reproductive process from the male ejaculate. Right? And the reason why, very good odds, one out of 150 million needed to fertilize that egg in the neck of the fallopian tube to bring about the entire human at the end of the pregnancy of the female. Let's have a look. So what we're seeing here, once you have the union of sperm and ova, now you have the formation of what is known as a zygote. Right? Both of them are haploid, so now you get a diploid number, or the full complement of the genetic material, and the cell begins to divide. Now let me ask you this. How many cells does a human have? Approximately, in a human. Let's say an adult, right? We're looking on the order of trillion, right? So approximately what will be a good average is, let's say, 40 trillion cells. Now, when we say trillion, what do we mean? Well, a million is 6 zeros, a billion is 9, so a trillion is 12 zeros. And all of that comes from just the union of two cells, a sperm and an ova. Absolutely mind-blowing. Do you all find that absolutely mind-blowing? If you find it mind-blowing, just raise your hand. All right. I just find it absolutely phenomenal when you think about it. Here it is, you have just two cells, and ultimately those two cells, they form 40 trillion by continued replication and dividing. From two, you have 40 trillion, and you have a complete human. OMG, that is absolutely incredible. And if I were to ask you, what is the size of a sperm cell? How big is it? Right? If you were to think about it, is approximately, in terms of the diameter, like 5 micrometers. And again, what is a micrometer? Remember, take your finger, your thumb and your forefinger, put it together. Now pull it apart. When you now start to see light between the two fingers, that distance is a millimeter. Divide that into 1,000, you'll get a micrometer. So imagine just 5 of those little 1,000th of that space, right, is 5 micrometers. And from that unites with the ovum, replicates until it reaches 40 trillion cells. And boom, it is absolutely mind-blowing. All the instructions written in those 23 pairs of chromosomes, which are in the nucleus of the cell. And it tells the cells, look, let's divide. Not only do we divide, but okay, you become a hand, you become certain layers of the body itself, and all of these instructions are written in something that you can't see. That is just absolutely mind-blowing when you think about it, right? This is on the order, imagine, micrometers, right? That all the instructions for a complete human being is actually written in there on those 23 chromosomes. That is just mind-blowing. It really is, all right? Or at least I think it's just totally mind-blowing. Let's move from mind-blowing. Let's get back to the fetus. So here we are seeing, in terms of repeated division, 2 becomes 4, becomes 8, 16, and you have ultimately, growth leading then to the development of the embryo. And then of course, you are shown here, right? So this successive division, leads to the embryonic development and ultimately fetal development, and ultimately to the embryo being formed as shown here. So one of the things, yeah, this is actually is pictured back to front in terms of this, yeah, the mirror image, because we start on this side and we go this way, right, in terms showing the replication, then development of the embryo, and ultimately the fetus. So here it is, we're seeing, this is a showing at the 3-month development stage here. And if you will notice, you have the eye, the air in terms of development, ribs, spleen, the umbilical cord, yolk sac, which provides nutrients until you have full placental development. And then you have nutrients which cross over from the mother to the developing embryo. Of course, once the embryonic period is finished, fetal development continues. Now, does the blood of the embryo normally, does the blood of the developing fetus and maternal blood, do they mix? Normally, do they mix? In other words, then how do the nutrients move from the mother to the baby? Is there like, one continuous from the placenta, it just continues on through the umbilical cord? Well, how does that work? How does the baby get the nutrients from the mother, right, once the placenta is fully developed? Anybody wants to hazard, I guess? One word. It begins with D. How the nutrients move, and it's a principle associated with movement of things.
Diffusion, sir.
There you go. Diffusion, yeah. So it diffuses, right? Very importantly. So there's no mixing of the blood normally. If you do have some kind of trauma and you have rupturing of the blood vessels, well, yes, you could have mixing of the blood, but normally the blood are separate types, right? And they're kept separate from the placenta, but the nutrients, carbon dioxide movement out of the fetal circulation into the maternal circulation, oxygen diffusing into the fetal circulation, all of that is done by diffusion, but you don't have a direct mixing of the maternal and fetal blood happening. All right. So in terms of the early development, we mentioned union of the two, of the male and female gametes, male being sperm, female ovum, leads to the development of a zygote. And the zygote divides by mitosis into identical cell called blastomeres. And they keep dividing until you have a blastocyst cavity, trophoblast, inner cell mass, and then you have implantation into the endometrium or the inner lining of the uterus. Let's have a look at that diagrammatically. So here we have the zygote, which is the union. So the sperm penetrates the ovum and unites with the nucleus of the ovum, and then by mitosis, the zygote divides and forms two separate cells, and it just continues. Mitosis, very importantly, mitosis produces identical cells, so you have continued division by mitosis. Ultimately, by day 3, it represents something that looks like a bunch of grapes, and hence the reason they call it a morula, which is really from the Latin for grape, because it looks like a bunch of grapes after day 3. And day 5, they keep dividing, but interestingly, the cells now begin to move away and they form a rim plus a dense inner cell mass with its fluid cavity. And this happens by day 5. And then at this stage is known as a blastocyst. So already from the simple union of sperm and ovum, we have division occurring and we have development, most critically, happening as well. So this inner cell mass, the amniotic cavity forms within it, and the cells closer to the cavity differentiate into hypoblasts, and the yolk cells, the yolk sac, takes the place of the blastocyst cavity. So you have then this clear differentiation happening. Right, and then you have implantation as a uterine wall, and now you have distinct layers being formed, hypoblast and an epiblast being formed as the division. So not only are you having division, but you're having within the cell mass, you're having specific layers also showing or beginning to differentiate within the egg itself. So the embryonic plate, this is a mass of cells, and now you begin to see distinct layers, an ectoderm, a mesoderm, and an endoderm. So here we are seeing, right, the hyphoblast and the epiblast. As time goes on, you have these distinct layers, an ectoderm, mesoderm, and an endoderm forming. The ectoderm forms the outer tissues, such as the skin, whereas the mesoderms forms the middle layers, such as tissues, such as the muscles, whereas the endoderm, it replaces the hyphoblast and forms the inner tissues, such as the lungs. So not only are you having these cells actively dividing, but you're also having them differentiating. They're becoming very specific, and in their specificity, they also are now beginning to develop into particular layers of the body. And in their development into these layers, they form certain structures which are associated with these layers. So this is just showing, right, the different layers here. So remember, initially you just had the two cells, but now you're having a mesoderm, a middle layer of cells, endoderm, an inner, and an ectoderm, the outer layer. And from this, you have, as mentioned before, the different developments of the different parts. Let's have a look at those parts which are developed. From the mesoderm, you have cardiac muscle, skeletal, the tubule cells of the kidney, red blood cells, and smooth muscle in the gut. Very important, smooth muscle. They are associated with the movement, peristaltic movement throughout the gut. The endoderm, the internal layer, that develops into the lungs, cells of the lungs, the thyroid cells, and the pancreatic cells. And finally, the ectoderm, you have the skin cells, the neuronal cells, and the pigment cells of the body. They actually developed from the ectoderm. So it's really incredible when you think about it. Yes, you have the union. Yes, they begin to replicate, but then you reach a point when not only do they replicate, but they become distinct layers. And these distinct layers, they keep replicating, and they ultimately develop into these structures, which we know are present in the complete, well, at birth in the baby that's formed. All right, so mentioned there, union of the sperm and ovum, forming zygote, then differentiating, forming a morula, forming the blastocyst, and then the blastocyst, the layers becoming specific, mesoderm, endoderm, and ectoderm, which then develops into the different structures of the body itself. Let's just talk about how they do that. It's all about your genes. That is the information which is encoded on those 23 pairs of chromosomes. Let's see if we could talk about those things. So we have 23 pairs of chromosomes, and the genes, very importantly, they're segments of DNA which code for certain proteins. Right, so that's in terms of you have several definitions. That's a very a widely accepted definition. And these proteins cause the expression of certain traits. Of course, we often wonder why it is we look at our parents, why it is, we have curly hair, that runs in the family and so on. So these certain traits, they all have to do with your genes. And alternate forms of a gene, they're known as alleles. So you have these genes which are present on the chromosomes. These genes code for particular proteins. You have alternate forms of those genes, and that is known as an allele. Collectively, all of these alleles, which forms all of the information, these are known as a person's genotype. Right? So the genotype refers to the alleles, and these are the genes. The phenotype are the physical expressions of the genotype. So in other words, then, the information written down within the genes, collectively, that's the genotype, but the expression of those genes, what you actually see, such as curly hair, such as brown eyes, right? Such as a straight nose, all of those things, those are phenotypes. Phenotypes, the physical expression of the genotype. So here we are looking at the chromosome. Let's have a look. So within, here's a cell, a human cell. Within the human cell, 22 autosomes and 1 pair of sex chromosomes. I forgot, in terms of males and females, how is a female represented and male in terms of chromosome, XX and XY? Which one is male, which one is female?
Male is
XY.
XY.
Male is XY and female is XX. Yeah. One way how I remember it, I think I share that with you all, woman is boss. As we all know, women run things. So they have more genetic information than males. And if you look at an X, if you remove this part, you know, if you just take this off, you're left with a Y. So genetically, men literally have less genetic information. So that's why I remember XY is male. And with the female, more information. So they have this part being present. So XX, if you think about it that way. On those 23 pairs, right, we have the chromosomes. So here it is, if you unwind, and they are tightly wound. Why is they are tightly wound? You know, if you look at it, and the strands, they're wound around proteins known as histones, Histones. You know, very much like you know, you have if you're flying kite and you have string, I don't know how many people fly kite, and you wrap it around a tin pan or what have you not. So that tin pan is very much like the proteins present in the chromosomes, and you have then the strands of the chromosome, the chromatids, wrapped around the protein. Now, why do they have them wrapped around? Why didn't they just leave them straight? So why do they have them wrapped around the proteins, the strands? For practical reason. Why are the chromatids wrapped around, you know, the proteins? So they're coiled around. And the same reason, when you think about your brain, for instance, as well, you know, your brain is coiled up, you know, really ruffled. When you think about your digestive tract, right? Remember, your small intestine, about four persons long, right?
So is it coiled because of the length?
Yeah. Yeah, so you can wrap up more information, quite right. If they had them straight, they'll take up a lot of space. So they have them coiled because when you have it coiled, something coiled or compressed, you can have a lot more in a very small space. And that's why it's done. So here it is, they're coiled up, as I said, around proteins known as histones. And these strands, it consists of these strands, which are known as DNA, right? Deoxyribonucleic acid. And the DNA is a double helix, and you have the bases, and outside you have a sugar phosphate backbone. So all along here is the sugar phosphate backbone, and you have the bases within running inside, like rungs of a ladder. Think about you have a ladder made out of gummy material, you know, so you could twist it. So you have the ladder straight and you just twist it, keep turning it, so you form coils. And the rungs of the ladder, or what you step on the ladder, those are what are the bases in the when you're looking at the chromosome. You have particular base pairing, and the person who figured that out, Shergoff et al., yeah, they figured they got a Nobel Prize for it. Adenine with thymine, guanine with cytosine. All right, this is in DNA. All right, and this is what a certain segment of this chromosome, this would code for a particular protein. Right, so a particular segment, and this particular segment which codes for a protein is known as a gene. So the phenotype shown here, blue eyes, and the genotype, small b, small b. This is recessive. When we are looking at the genes, a dominant, as shown here, dominant allele or alternate form of the gene is written with a capital B. The recessive form of the gene is written with a common B. So when it's recessive, if a dominant is present with a recessive, the dominant is always shown, the capital B, whatever trait that is. So in this case, brown eyes is the dominant trait. So if you have a dominant, big B with a small b, the result will be brown eyes. The only time when something is recessive that you will have it actually showing is when you have two recessive genotypes. All right, and in this case, you have blue eyes. So allele We mentioned the alternate form of a gene, and also we also mentioned in terms of the dominant is written by a capital and the recessive is written by a lowercase letter. And they could be located, this is just showing, this is a chromosome, and the loci, the gene loci or locus is where it is found on the particular chromosome itself specifically. All right, so it's like an address in terms of the locus, and it tells you where you would find this particular gene on the chromosome. All right, so for the alleles, for the alleles themselves, you have homozygous and heterozygous. Homo, the prefix homo means same, and hetero means other, not alike. So the homozygous, these are genotypes with two dominant alleles, so they have the same, either the dominant form, biggie, or the recessive form, but they are both the same. Homo meaning same, so the two forms of the allele, they are the same. Hetero means other, or like not the same, so therefore you'd have two which are not alike, they are not the same. So we have a big E and a small e. Since this one is dominant, whatever trait is represented by the capital letter, that is the one which will be shown in the phenotype. Now the guy who figured this all out, who was the person who is often referred to as the quote unquote father of genetics? His name begins with M and it rhymes with Zendel. He was a monk, and his name was Gregor Mendel, yes. Yeah, Gregor Mendel. Now, oftentimes people say, why did he use peas? If I were to ask you that, why he used peas? What would you say? Why he didn't use Karaili? Why he didn't use cashew? Why did he use peas? Anybody? Well, interestingly, back at the time when he was around, peas was a big thing, right? Peas was the cash crop. So persons who have peas companies, they wanted to figure out what's the best way, to get smooth peas, because that was the one that demanded the best prices. So it didn't have custard back then, and it didn't have gate. So persons is either you were very wealthy in terms of going to get education, or you had somebody sponsor you through school. So these peas companies, they were like, look, we need to figure out on a consistent basis how we could get these smooth peas to sell. So literally what they would do, right, they would scout around and, look for individuals. All right, let me see, who's showing promise. And then they would send them off to university. And these universities back in the day, as I said, it didn't have Costart and UWI, but the universities or the places of education, it was in the monasteries, right? So therefore, to become educated, you had to go through not only the education process, but you had to become a monk as well, go through the religious process. So hence, enter Gregor Mendel, right? He went through, and the reason why he chose peas, as I said, is because it was a cash crop at the time. And he did his research on it, and he was able to show then that, using certain crosses that you could predict the outcome based on the certain type of peas that you use. You could, if you crossbreed them, get certain outcome. He actually was able to predict it, and he wrote, a big paper, and he wrote research on it, about his research. All right, so all of that in terms of Mendel's work could be compressed into what we know as a Punnett Square. And what the Punnett Square shows is the likelihood of passing on a trait. In the case of with Mendel, it was peas, and he was looking at rough or smooth, and also the color of the pea. That also determined the price. So he was looking at those particular traits. But when you look at humans, you could also have different traits. All right. So these traits, they're represented as the genotypes from the mother goes on the top and the father goes on the side. Let's see what we mean by that. So for the mother, right, so it goes across and the father comes down in terms of the inheritance. So this one is known as dihybrid inheritance. It's showing two particular alleles or alternate form of a gene. And so for the mother, with this mother, what would you say she is? Is she heterozygous or homozygous? It's a big C and a small c. So is it
The mother?
homo? Yeah,
Hetero.
hetero, right? Because it's a big C and a small c. This is the dad down here. Big C, small c. Is he homozygous or heterozygous?
Hetero.
Hetero, yeah. Remember, homo means same. Now, out of all of these four, 1, 2, 3, 4, which square has homozygous? Actually, if you read really fast, you'll figure it out. Which colors represent homozygous dominant? Which color represents homozygous dominant?
The pink.
The
Purple,
pink square.
pink, sorry, the purple.
Pink
yeah.
or purple, depending on your screen, right? So this one is the homozygous dominant, and the homozygous recessive down
The
here.
one out of Julia.
Blue.
Yeah, Julia, right? So there's the homozygous recessive in terms of the particular disease that is passed along. So how do you determine it? This is how it's done. So you write this is known as a Punnett Square. So like tic-tac-toe in a kind of a way. And you just write, you separate the alleles or the alternate forms of the gene. So in this case, we're talking about dihybrid inheritance. And here you have the big C, small c, and you separate them, right? So you put the big C here, small c here. You do the same thing for the father. He's heterozygous, you put the big C, small c, and then you just come across. So you come across here, you write the father, the mother. And in terms of convention, you always write a capital letter first. So let's go. One from the, so you come across here, the father would give a big C, and coming down, the mother would give a big C, you say write it next to it, and a small c. Under here,
Excuse.
Yeah, go ahead.
You write them? Because I'm not seeing. I don't know. If
Oh no, I'm not writing. You all seeing the arrow?
No.
You're not seeing
no,
the arrow?
sir. No.
Oh, no wonder. And look at how I joyfully. Okay. You all seen the dot?
Yes, sir.
Okay. So from the mother, right, you have the big C, small c. From the father, big C, small c. And only do you write father, you come across for the father. So the father now would be big C, and coming down the mother would be big C here, and the other one will be small c. Coming across here, the father will be small c. The mother, big C, so you bring it down, and you small c. And by convention, you always write the capital letter first. So at the end of the day, what you end up with, one homozygous dominant, one homozygous recessive, and two heterozygous, right? And here we have, so we are looking at one allele. Now we're going to look at two. Right? So the first one, when you're looking at one particular trait, monohybrid inheritance, this one is a dihybrid inheritance. And you do the same thing. So the parent in this case, right, it's heterozygous, and you just separate them accordingly. So for this one, it's two traits you're looking at. So the possible combinations, big R, big Y, big R, small y, small r, big Y, small r, small y. So you just write those combinations here, and you do the same thing for the other parent, which will be the male. All right, so the big R, big Y, the possible combinations, big R, small y, small r, big Y, small r, small y. And you just write them down, and then now you just join them. So bring this one over here, bring this one down, bring this one over here, big R, big Y, big R, bring this one down. And then once you have them all here, you do all the combinations, big R, big Y, small r, small y, then you just arrange them as per the convention, where capital letters go first and the common letters follow it. But that's all you do. So you just bring this across, this will be coming across on all of them. So you take the big R, small y, put it here, 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 and here, and then you come down. Take this and you put it here, this one next to it, this one next to it, this one next to it. And then when you do that now, you rearrange as per the convention with the capital and the common letters. And then you figure out, you do it for all, and then you would see all of the possible combinations. And usually for a dihybrid heterozygous, you get a 9 to 3 to 3 to 1 ratio. Right, so you'd have 9 heterozygous, 3 where you'd have the homozygous dominant shown here, 9 to 3, 9 and 3, 12 and 3, 15 and 1, 16. So you'd have 9 to 3 to 3. And you would have just one where you'd have the homozygous recessive being present, right? And how did they figure out it was 9 to 3 to 3 to 1? How did they know this would work? Well, they actually did it for a number of plants, and they found consistently when they did these crosses, looking at this particular trait, that yes, you always consistently got in the offspring, you got this representation of the P's in this. So when you're looking, we're talking, this was done repeatedly on a number of different tree plants, and all the time when they looked at the P's, this was the outcome. We're not talking one, we're talking about thousands of plants, but the ratio would always be in this 9 to 3 to 3 to 1. So that's how, using that data, they were able to come up with this Punnett Square. And this holds true for dihybrid when you're looking at 2 particular traits represented when they are crossed in terms of the heterozygous form. All right, this could be carried over in terms of with humans as well, which is known as pedigree. And this is used, pedigree refers to the mapping of the inheritance of a genetic disorder or a trait. So in this case, the mother is a carrier and the father is a carrier. So in terms of the offspring, same thing, half. So you break them up into half and half, half and half, and then you join them up and you will get this type of situation. So from the mother, if the mother is a carrier, in terms of the outcomes, right, you'd get a normal, John a carrier, Justin, who is normal, and Julia, who is, well, in terms of this particular disease, if you're homozygous recessive, it's lethal. So then this case, this scenario, this one would be lethal. All right, so as related to these genetic inheritance, you also have some disorder in the inheritance patterns. So sometimes you get certain inheritance which don't follow what is known as typical Mendelian inheritance, which is what we saw with the Punnett Square. And with some of them, you have, this is due to autosomal disorders, which are disorders located on the autosomes. Autosomes, those are the 22 pairs. Remember, we have 22 pairs of autosomes and 1 pair of sex chromosomes. So the autosomes are all the chromosomes except the sex chromosomes. You have an autosomal recessive in your individuals with a disease, a homozygous recessive. So they both inherit each of the alternate forms of the gene from a parent, and the carriers are heterozygous. So which is normally, when we think about the Mendelian inheritance, right, is this one is a little different to that. When we look at the autosomal dominant, individuals with the disease are heterozygous and homozygous dominant. For the heterozygous dominant only, there's a 50% chance of having a normal child and a 50% chance of having an affected child. Let's look at that diagrammatically. So here, the father is a carrier, the mother is a carrier. So in terms of the outcomes, so this is again carrier, so therefore there's a half chance of having the affected disease carrier. So this is shown here. So half would come from here, half from here. So you have a fully affected child. Half could come from here, half from the normal. So you have a carrier. And then of course, you could have an unaffected child as shown here. So this is just reinforcing same thing what we just looked at. So the X-linked recessive, these are disorders encoded by genes on the X-linked chromosomes. So we're talking about the sex chromosomes now. Most X-linked chromosomal are from the X chromosome because half of the population doesn't have a Y chromosome, right? In terms of male and females, which is much smaller than the X and doesn't contain as much genes. That's why most of them are X-linked. Most of these X-linked disorders occur in males. All right, let me just have a quick look at the chat. Right. So in females who have two X chromosomes, an abnormality on one is typically compensated. So if you do have it on one, the other one is normal. So therefore, let's say it's some particular protein that is not being manufactured, well, the other chromosome will actually compensate for it, right? Because it will be able to produce that particular protein. Most common example is color blindness. So males are usually more afflicted because while you do have a pair of chromosomes, and in terms of that particular gene producing the protein, which prevents color blindness, let's say if the person does have, is afflicted or doesn't produce that protein, which prevents the color blindness in males, well, in terms of the other, because with males, they don't have that part of the chromosome, right? They have a Y, so they're missing part, so therefore they cannot compensate for it in terms of the other chromosome producing it, or the other strand of the chromosome producing it. So therefore, what would happen? They would show the trait more readily than with females. With females, for them to show the trait, they need to have both chromosomes actually having the defect for them to show that particular trait. And it's shown here in terms of with color blindness, because it's X-linked, shown here. Because the males, they lack that bit here that actually, so once it's present on one, it will show the trait. So for the offspring, right, you'd have 2 males, 2 females, but the X should have one. If the X, this comes from the father, you'd have a son that is colorblind, which is the Y, shown here. The X daughter that is, if the 2 X's, 1 X from here, 1 X from here, you'd have a daughter that is colorblind. You have 1 X here, 1 X here, 1 X here. You'd have a daughter that is a carrier because of the fact that there's another gene, right? So let's say there's a particular protein which is responsible. When it's not produced, it causes color blindness. All right, so with the affected person, that particular protein is not being formed, but hey, on the other chromosome, that could actually produce it because it has the same gene locus, so it could produce it. With males, they don't have that luxury because they're missing that part, a particular part of the chromosome, which forms the X. So once they do get it on one chromosome, and that one chromosome is not producing that protein, they will show the trait. So in this case, the son is colorblind. All right, so what did we look at today? Well, we looked at inheritance patterns. We looked at the amazing world of humans in terms of how this inheritance from 2 chromosomes could then lead to the formation, of 2 cells, one of the sperm, one from the ovum, uniting to form a zygote, ultimately replicates to form 40 trillion cells, which we all know as a complete human being, which is absolutely incredible. 150 million sperm per ejaculate and 5 milliliters of semen. Why so many? Because it is important. Without it, we would disappear as a species. Once you have this union of the two gametes, the male and female, forming a zygote, replication continues. Initially, in terms of the replicative process, or very important to note, it's a mitotic process. So the division, it produces a blastocyst, morula, and then not only do you have the division, but you also have specialization. You have the formation of specific layers, a mesoderm, endoderm, and ectoderm, which then becomes the skin, becomes the lungs, becomes the blood. They then as further differentiation occurs. All of these things nicely encapsulated because of the fact the instructions are written on those 23 pairs of chromosomes in our cells. And that information that is written there, it could be predicted in terms of getting certain outcomes relating to disease and the disease states. And that is nicely done using Punnett Square. So we have with a Punnett Square, you put the genes, the alternate form of the genes, right? Alleles, you write them by convention, the mother goes on top, the male goes down on the left side, and then you could do a comparison with the monohybrid inheritance. You could do it as that, and you could also do it with dihybrid inheritance when you're looking at two particular traits, and which you will get a 93:3:1 ratio with the dihybrid inheritance, which then shows that the particular traits are passed on. Not only could it be seen for two traits, but they are also associated with disease states and X-linked, very important to note, X-linked traits that these do not follow typical Mendelian genetic inheritance because of the fact that you wouldn't get the same with males, in terms of the inheritance on the X chromosome, because of the fact that they are missing part of that chromosome. Once it is present, they will show the disease, whereas with the female, they will actually be carriers, and only when they are homozygous for that trait will they actually show the disease state. Okay, and that is where we'll stop the lecture for today.